Okay, so thank you guys everyone for coming and joining us virtually for our Wild Lecture series. This is the first one of the year, so you guys definitely have made an awesome attendance so far. And today we're going to learn all about Morgan Slevin and his presentation on Northern Cardinals and his research. He is a PhD candidate with FAU and definitely has a ton to teach you guys about Northern Cardinals and all of his research. So I will go ahead and pass it off to him.
Thank you so much for having me, Jordan. So yeah, as she mentioned, I'm working on my dissertation at Florida Atlantic University. So this is part of that research. And I'm trying to figure out how the bacteria in the gut and other various parts of a bird are related to its health and a couple other things that are important. I want to start off by saying this has been a collaborative, highly integrative study. I have a lot of people to thank, starting with my my advisor, Randy Anderson at Florida Atlantic University, and then also some collaborators up in New York. I've got Jen Hoots and Marian Vatusik at Cornell University, and Dan Baldassari up at SUNY Oswego. So huge thanks to them and to South Florida Wildlife Center for having me. Y'all do such great work pretty much every single day for these animals. You save so many things, and I'm hoping that my research can help add to that, a little bit down the line, and find some long-term solutions for some of the stuff that we will discuss today. So let's get into it. Starting, what is a microbiome? You've probably heard the term tossed around a little bit, and if you're going to understand the talk today, we need to have a little bit of talk about what that might be. So micro means small, biome means community. So it is a small microscopic bacterial community composed of bacteria and other microbes. And the first thing that people think when they hear bacteria is probably, infection. But bacteria doesn't always mean infection. In fact, these communities are composed of mostly neutral bacteria that do a whole lot of nothing and just take up space, also some good and some bad bacteria. So there's more to meets the eye than just, infection and pathogens. And so that kind of describes what a community is, but where are these communities? Well, they're everywhere. They're all over you. They make up probably over 50% of the cells in your body. And the way that they can be so widespread is they actually serve a lot of different purposes. In your nose, they help with mucus production and antimicrobial chemicals. They assist in digestion in your mouth, and they have all sorts of other functions throughout the body. The biggest one is probably in the colon. That's the most focused on because it aids in digestion and also has some other characteristics that we'll discuss in a bit. But they really do have a function pretty much everywhere, and they are everywhere. But what determines what sorts of bacteria are in that community? Well, there's a lot of different filters. Namely, genetics is the big one. Two humans will have very similar microbiomes, whereas a human and, say, a dog will have slightly different microbiomes. But the human and the dog will have two microbiomes that are similar to one another compared to a human and, say, a fish. But environment matters a lot too. The goldfish on your desk might have a different microbiome than a fish at the bottom of like a deep sea trench. So environment for the host matters. Microclimate in that bacterial community is really important. The pH, the oxygen content, amount of sunlight, temperature, it all really matters and determines what sorts of bacteria are found in that community. And what sorts of bacteria are found in that community determine the functions that you see here to the right. So it's important to understand this holistic list of determinants when understanding and studying microbiomes. So humans like our brains. We love our brains. We're really proud of them. We have a big ego when it comes to our brains and our intelligence. We think that's what sets us apart from other animals. And it turns out there's a lot of emerging connections between the gut microbiome, which is the microbiome in the gastrointestinal tract of humans, and a lot of aspects of cognition, including learning and memory, vocal communication, a whole suite of behaviors that define mental health, like depression and anxiety. And so all of a sudden, humans care a whole lot about microbiomes. There has been a lot of money invested into this field because humans want to know how to make the thing we love most better, or at least keep it from not doing so great. But it's not just the brain that matters to humans. We care about our overall health as well. And other things that we specialize in is stress. Humans definitely specialize in stress. And this is one of the other indirect ways that the microbiome in the body, in the gut, can impact our brain. So the brain is affected by a lot of different things in the body, and a lot of different things affect the brain. The stress response is a good example of this, in that it's actually an evolutionarily good thing. So we have evolved the stress response to cope with the things that cause the stress in the first place. If you're getting chased by a bear and you are choosing to fight or flight, hopefully you're going to run away. You need the energy reserves to actually run from that bear. And that happens through a series of cascades coming down through different parts of your brain and ultimately to your adrenal glands that release glucocorticoids, which are the hormones that actually mobilize these energy reserves. And all of that is called the hypothalamic pituitary adrenal axis, or HPA axis for short. And so this sounds like a positive event. It is very good, but one of the negative offshoots of a stress response is an immune response, which also sounds pretty good, right? You're fighting off the bad bacteria, but that can actually have, it can just indiscriminately kill a lot of bacteria in the gut microbiome and other microbiomes. That can open the door for opportunistic pathogens to move in, and that can alter signaling with the vagus nerve with the brain. So this whole holistic series of interactions is called the microbiota gut-brain HPA axis, and with the health of the host lying at its center. Now, this total mouthful, complete mouthful of concepts is pretty complex. You can see it takes up the whole system of the body, and we know a decent amount in humans and rodents, because we use lab rats as a way to study this axis in humans, but we don't know a whole lot about that in other animals. And so that's kind of the focus of my dissertation. So despite these exciting advancements in rodents and humans, we still know relatively very little about the microbiota gut-brain HPA axis in other animals that I particularly care about. I like humans. I am a human. I care about them. But ultimately, I want to focus on the wildlife. That's what my background is in. That's what my future will be in. So there is actually a whole lot that they could stand to benefit from us better understanding the microbiota gut-brain HPA axis. And one of the biggest ones is stress. So I've described the ways that stress can wreak havoc on the body and how it can wreak havoc on the microbiome. And that's actually kind of what brings me to this talk today, is South Florida Wildlife Center. They care for so many animals, thousands per year, and nearly every single animal, they have to battle stress. Because when an animal is being cared for by a human, it doesn't think like, oh, I'm being saved. No, it just thinks that it is about to be eaten or killed, no matter what sorts of measures the vets take. No matter what measures the veterinarians take, the animals are always going to be stressed. That is just what happens when wildlife are in human care. And there's no way around it. They take so many awesome measures to help reduce stress in these animals, but sometimes stress can lead to treatment failure. And it can impact appetite, it can impact behavior and grooming and self-maintenance, and it can even slow the process of healing. So if there's some sort of way to mitigate stress and really ameliorate that sort of negative effect in these wildlife, there's huge applications to this, and it extends the conservation efforts of captive breeding programs with endangered species. Florida has a lot of species that are not doing too great and might be in captive breeding programs or already are. And so I chose to try to illuminate and figure out what is going on in this gut-brain HPA axis. And I chose birds because that's what my background is chiefly. But there's also the fact that we already know a whole lot about the brain and about stress in birds. It's very well studied. There are books and books and books written on this. Not a whole lot known about the avian gut microbiome, though. So that's where I and a bunch of other people are hoping to come in and change that. So how can we study avian microbiomes? How can you look at a microscopic community in a wild bird? Well, first you have to catch the bird so you can get your sample. And so we use what are called mist nets. If you look closely here, my colleague is extracting a tiny warbler from a mist net. A mist net is basically like a glorified volleyball net. It's nearly invisible. It's strung between two metal poles. And if we put a audio speaker that plays the song of whatever bird we're trying to capture, that will typically bring the bird into the net because it hears that song and thinks that there's another bird intruding onto its territory. And it thinks, what the hell's going on? It flies in, it hits the net, and then we very carefully extract it from the net. And then from there, it's actually really easy. That's probably the toughest part right there, because once they're in the hand, you can really, really gently handle them. You don't have to be constantly squeezing them and restraining them from getting them away. You simply just hold on to them in a proper way that we've all been trained to do. And then you can take a whole series of measurements and other samples to learn more about this bird. First thing we typically do is we put color bands on these birds. That way we don't have to usually catch them again if we want to figure out which bird is which. So I can release this bird, and through binoculars, I can see this unique combination of colors around its legs. And I can definitely tell that it's different from this bird, which has a different series of colors on its legs. And so I don't have to catch that bird again to know that this is bird number, such and such. We can take a whole bunch of measurements to assess body size, beak characteristic, and things like that. We can also take more complex measurements, like this microbiome sample I'm taking from the bird's mouth. And once we are ready, once we have everything that we need from the bird, we release it, and it's gone, good as new, back to its life. And if you look closely at the end there, you can actually see the little bands on the legs. So they really stick out, which is great. That whole process I have been doing for about 10 years now. I've captured close to 1,000 birds. I'm very experienced at doing this, and I can do it very safely. And if we couldn't, we wouldn't do this research. The whole purpose of it is to be able to study these animals, these wonderful creatures that are constantly around us, and be able to learn more about them so that we can hopefully save them one day if they need saving. So that whole process does sometimes induce mild stress. They picture it as a potential predation event. So they think that they might be captured and eaten, but then they're released and they recover back to their normal levels pretty quickly. And they can choose not to participate. I mean, if a bird is, right on the edge of its health and it's all too stressed out, it won't fly in the first place. So there's no way that we will, do irreparable damage to this bird, provided we do everything safely, like I always have been doing. So it's a really cool way to get all of these measures. And it's actually nice to have a fringe benefit of the capture being a little bit stressful, because that ultimately is the mechanism that I am studying and testing in my dissertation here. So we got the idea for this study, the one that I'm currently in progress on, and we'll be presenting some data on today, as well as we'll be continuing this year. We got the idea for the study, oddly enough, from a squirrel paper. Like I said, everything we know is from mammals. We don't know a whole lot from birds. So what these researchers found is that wild red squirrels that were captured and then sampled for their oral microbiome and sampled for their glucocorticoid levels, or CORT for short, which is that hormone involved in the stress response. If these squirrels were captured, released, and then recaptured again 2 weeks later, they had lower diversity and abundance of species in their microbiomes, and they had a higher abundance of pathogens in their microbiome, and their stress levels were significantly higher on that second capture. So you're looking at some metrics of health here, you're looking at some metrics of the microbiome, and it seems like they're a little bit worse off after that stress of initial capture. And so this is a nice way to really ask our questions of what stress does to the microbiome in a bird. So I want to emulate this in Northern Cardinals. Northern Cardinals are very common, widespread. They're pretty easy to catch because they are an angry bird. They're very territorial, which means they're much more likely to fly in when you're setting up a mist net and audio playback to lure them in. So they're pretty easy to work with. But they also have a kind of semi-unique characteristic to them, which is this bright coloration. That's what most people really love about this species, is they have this bright red coloration, and their beak is actually bright reddish orange as well. So that color is no accident. It's not just there, it's serving a purpose. And it's what we call an honest signal. So an honest signal is something where the animal is sort of conveying information. In this case, it is the quality of this bird, and it can't cheat or lie about that signal, because the pigment that makes this coloration is called a carotenoid pigment. And they can't just naturally synthesize this internally, just endlessly, and make redder and redder plumage. They have to get this pigment from their diet. And so they can't really cheat. You know, however good they are at getting this pigment, that's how much they have available to put into their tissues to make this coloration. And they also use that carotenoids throughout the body as an antioxidant in the immune response. So they can't just endlessly put it into their plumage. So they have to be honest about what they are signaling. In this case, it's their quality. The cool thing about their beak is, because that gets a blood flow, that color is vascularized tissue. And so it can update with depending on, you know, how their stress is fluctuating, how their health is fluctuating. And so that's a dynamic measure of health that I can look at while I am sampling these birds. And there's other studies that have shown already that female cardinals look at the beak color of a male when they are, you know, assessing him to see if he's a good mate or not. And males with more red beaks and, you know, more elaborate ornamentation and coloration, they're more often selected. So it stands to reason that females might actually be able to gain some sort of microbiome information from assessing the beak coloration of a bird, if the coloration is, you know, fluctuating with health, and if the microbiome is fluctuating with health. If it's all tied in together, then females could actually be getting some interesting information just by assessing this bird by looking at it. You know, they don't have to do any DNA sequencing like I do to figure out what's going on in the microbiome. So for my experimental design, I am capturing birds at Treetops Park. Some of you may be familiar with that. It's in Davie, Florida. Mostly forested, a lot of open area too, and a lot of good trails. Cardinals like to fly around at this place. There are so many cardinals at this place. So I've been working here since 2020, still working there, and captured a lot of birds. Every one that I catch, I put color bands on that you can see here. I'm mostly catching males for the study because they're the ones that are reliably territorial, but I'm also catching females and some young birds as well to see how the microbiome compares across these different demographics. For each male that I catch, I take a series of blood samples. So this is how I can assess their stress levels. The glucocorticoid hormone, CORT, in birds is corticosterone, or CORT for short, that circulates throughout the body in a typical acute stress response. And it takes about 3 minutes or so to really start pumping through those veins. And so if I can take a blood sample immediately upon catching the bird, and then take another blood sample about 30 minutes later, I can see the baseline and the after effect of, you know, what their response is to the stressful event of being captured. So I take blood samples to assess stress responsiveness. I take standardized photographs of the beak so I can look at ornamentation, coloration. I take the cloacal swab and oral swab of these birds, as well as fecal samples if they are presented to me. And then some body measurements as well, so that I can assess body condition, which is one of the best ways to really assess the fitness of this bird and, you know, their current health, just based on how heavy they are relative to their body size. So how do we actually understand microbiomes though? It's this microscopic community. It's practically abstract. It's so small. How can you look at, you know, diversity and other ways to, you know, measure its health and its goodness? Well, we use diversity because that's one of the things that, you know, we already know a lot about when we look at, you know, the ecology of larger ecosystems like these flower beds that you see here. So alpha diversity is the first and simplest diversity metric that we use. It's essentially a measure of how many different types of bacteria or organisms broadly are in the community. So it's the richness of the community. So you can see that this community on the left has one type. As robust as this community is, you know, beautiful, beautiful flowers, but there's only the one type of plant. Whereas the community on the right has many types. It has a higher alpha diversity, many different types of organisms. We apply those same rules to bacteria, and we count the number of different types and some of their phylogenetic relationships to assess diversity. So that is the diversity within one sample. It's also important to be able to group samples together and compare them among, say, treatment groups between males and females, something like that. And so for that, we use beta diversity. And this is a measure of community composition, and it's our way of assessing similarity between samples. So you can see that the alpha diversity of these two communities is probably about the same, but I'm willing to bet they have largely different functions. The one on the left might be kept for greenery and ornamentation and things like that. It could be used for gardening, whereas the one on the right might be great for pollinators. So you can't really say that one is better than the other or worse than the other. We just say that we classify them as different. This one is different from that one. And then you can zero in on what specific members of that community and what their abundance is that might drive the kind of the function of the community. There's a lot that goes into it though, just to go from that swab to this is our number for diversity. So for the microbiologists in the audience, I will briefly go through it. Feel free to ask me any questions at the end if you'd like. But we have to extract the DNA from each swab sample that we take, amplify a region of interest that will help us differentiate between the different species of bacteria. And then we sequence those bacterial DNA sequences. That tells us what the actual DNA sequences are of the samples that we have obtained that make up that community. And then once I get my raw sequence data back, I do a whole lot of cleaning, filtering, and organizing, and I'm able to actually assign those DNA sequences to what known bacteria each one is made up of. And then from there, it's more filtering and normalizing and decontaminating. And ultimately, I can get my alpha and beta diversity metrics here, which are the real numbers that I'm using in my analyses. So this is an ongoing project. And right now, I will be presenting the data from my 2020 field season. My 2021 samples are in the works, and I hope to get those back soon. So for now, we have about 200 total samples. Most of those are cloacal and oral swabs with some fecal samples, and then of course negative controls so that I can assess decontamination. Perhaps the coolest part of this work is that I got to actually do some work at South Florida Wildlife Center and collaborate with them with getting some microbiome samples. So, all of this is driven by fixing animal health and finding a way to help out these wild animals that are so stressed out when they're trying to heal. And so the center gets cardinals. And so I've been working with some vets to have the vets and their staff take cloacal swabs of these birds. I was even able to come on site and take a couple myself, as well as any resident patients that they have. So it's a somewhat smaller sample size than the wild free living birds at Treetops Park, but it's been a really interesting look into how their microbiomes might be different. So looking first at Shannon diversity, which is the measure of alpha diversity that we use. Remember, that's community richness. That's the number of different types of bacteria in a single sample. It seems like Treetops Park might have slightly more diverse samples than South Florida Wildlife Center birds. The stats reveal that is not to be the case, which is just fine, but the sample size, I only have 12 birds from 2020, and then another 30 or so are going to be going into my current sequencing run from 2021. So I'm really hoping that a bolstered sample size will help clarify if there is or is not an actual difference in diversity between these different groups of birds. Because alpha diversity is probably the easiest thing to really understand and interpret when it comes to, is there or is there not an issue with the microbiome when they are healing at the rehab hospital. A little bit more complex is the beta diversity. So just to understand the graph that you're looking at here, remember that beta diversity is a measure of similarity or dissimilarity. And so the closer 2 points are on the graph, that those 2 points communities based on structure, based on abundance of what types of bacteria are there, the closer those 2 points are, that is, those points are more similar to one another. The further away 2 points are, the more dissimilar they are. So you can draw some ellipses around these to organize them by location. So the darker points are from South Florida Wildlife Center. You can see that they do seem to occupy a different part of the graph overall, at least based on the centroid of this ellipse. And the stats reveal that there is a significant difference in beta diversity between the two different sampling locations. So that tells us there's a difference. It doesn't really tell us what those differences are. So if we dig a little bit deeper, we can look at the specific genera that are present in each group of samples. And we can start to pick apart some trends here. So this is a graph of relative abundance, meaning of the total 100% of the bacteria of all the communities averaged together for one group, whether it's South Florida Wildlife Center or Treetops Park. This is the breakdown of what types of bacteria make up that total abundance. So I want to draw your attention to some of the bigger changes or differences between the two groups of birds. So Clostridium, Enterococcus, and Escherichia, they seem to be, they typically are genera that have a lot of pathogens within there. And so it's looking like South Florida Wildlife Center birds have a higher abundance of pathogens compared to free living birds. And just to be absolutely clear, I'm not saying that they're going to the rehab hospital and they're getting sick. What I'm pointing out that their immune system is taking a hit while they're healing, because that's just what happens. If you ever think about how stressed out you are, if you always got sick around finals time, that's because your body is going through something because you're really stressed out. And so your immune system is going to take a hit. Same thing happens when they are injured and need to be recuperating at the hospital. So it seems like their immune system might be a little bit compromised, and they have a higher abundance of these more dominant taxa. Other prokaryotes I wanted to point out is any bacterial genus that makes up less than 1% of the total abundance. So we just lump them all together. And you can see here that there seem to be fewer dominant taxa in the South Florida Wildlife Center birds compared to those at Treetops Park. So there may in fact be some differences in alpha diversity as well here. And it's generally good to have a more varied, more diverse microbiome. If you have fewer dominant taxa, then you might have an issue if one of those is suddenly removed. You have a big gap in your microbiome, and then different things can move in. You tend to have more stability with a higher alpha diversity. So, time will tell, more samples will tell. We'll see if these turn out to be really important or not. So that concludes my data representation for the South Florida Wildlife Center birds in particular. I'm actually really excited to present that. This is the first time that the world has seen any of the microbiome data from the center. So, thank you to them for actually allowing me to get these samples and include them in my research. What follows here is my data from the free living birds at Treetops Park, from which I was able to take some of these health proxies. I was able to look at body condition. I was able to assess the beak coloration and everything else. I want to point out before moving forward, my stress data is not quite analyzed yet, so I will not be able to present the blood sample data in the talk today, but we can still talk about some interesting things with body condition and beak coloration. So starting off, we need to figure out, how does alpha diversity differ across the different microbiome types? Just because they're from the same bird doesn't mean they're all going to be the same microbiome. So remember, microclimate is really important here, and that's going to determine a lot of the diversity here. So it turns out there is a significant difference in alpha diversity across the 3 different microbiome types, with, it seems like, fecal having the highest diversity, followed by oral and then cloacal. But there is no significant difference between males and females. That's to be expected. Alpha diversity typically doesn't change all that much between the sexes in any of the other studies that I've done with birds. But here's the exciting result. Here's where it gets really interesting to me, is birds with better body condition had higher alpha diversity. This is, but that was only in the cloacal microbiome. So this shows a really nice link between birds that are in better condition and their microbiome characteristics. A microbiome characteristic that I might add that is usually like the first one that researchers look at, because it's such a nice broad overview of what's going on in that system. So just a reminder, body condition in this case is the mass of the bird divided by the length of the tarsus, which is its longest leg bone. So it's essentially a measure of how heavy is the bird relative to its body size. And so I want to point out that this is, there's a distinction to be made between causation and correlation. I can't say that these birds have higher, that they have better body condition because of their microbiome or vice versa. There's no causation we can really look at here without some more manipulative studies, but it does still answer my question of, can you predict the health of the organism by looking at its microbiome? So that's really encouraging moving forward for me. And here's where honestly, I was really surprised to be able to see this sort of result. The hypothesis was there, but I was still really happy to see it come to fruition. So alpha diversity was shown to vary with beak hue. So I want to give you the lay of the land first before showing the data. I want to remind you about honest signaling, that birds with redder beaks tended to have higher pigmentation and are expected to be of higher quality compared to birds with more orangey, paler beaks. And it turns out that birds with redder beaks had higher alpha diversity, but again, only in the collacal microbiome and not in the fecal or the oral. So this shows a really nice link between the microbiome and sexual selection, because it confirms my hypothesis that females may in fact be able to obtain some sort of indirect measure and assessment of the microbiome just by assessing the color of a male's beak, which they already do during mating anyway. So sorry, dog wanted some attention.
No worries.
So yeah. No, you're good. I'm back. Yeah, our dog wanted to be part of the presentation. So this is a nice link between both natural selection with body condition and sexual selection with BQ. And some bonus data here that I didn't mention I collected is we also took some feathers, and we were able to do some reflectance spectrophotometry and find out that males with brighter back feathers, so greater ornamentation, they also had higher alpha diversity in their collacal microbiome. So it's just piling it on. The more ornamented these males are, the better their microbiome is. The more successful they might stand to be in sexual selection. So yeah, I'm going to take a break from this kind of data heavy section, because I know it's a lot of numbers and dots and stuff. This is supposed to be general audience. So there's this graph that shows that there might be something happening in my study that yeah, so we'll see what's going on here, but this is an abundance this is a graph of relative abundance, and this shows the abundance of a bacteria, Rotheacristenic, and it is shown to be significantly higher in abundance in the oral microbiome of cardinals compared to that of collacal or fecal samples. So what is Rotheacristenic? Why do we care about this? Well, it turns out that as far as anyone knows, it is only found on the human skin, and it is, it may be present in birds, but this is the first record of it being anywhere but human skin. So you might be asking yourself, how is it that this could happen? Well, it seems like I might be adding some to the oral microbiome of these birds, because they tend to bite anything. And they usually bite me before I can get that oral swab. So I found this kind of hilarious that this actually showed up in the data. This could all be nothing. This could actually be just a common bacterial type in the oral microbiome that no one has described yet, because no one has described the oral microbiome of birds. This is actually only like the third or fourth study to look at the oral microbiome of any species of bird. So I am actually really interested to see, hilarity aside, if this is an actual member of their community or not, but they will bite just anything. So it's fun to work with these guys and gals. So back to the feature presentation. Moving on to beta diversity now. So remember, beta diversity is a measure of dissimilarity. And this looks at relative abundances and the phylogenetics of the different bacteria that are there. And you can see here, and if you focus on the stats as well, there is a significant difference across the three different types of microbiomes in their beta diversity. And this is perhaps the most expected result of the entire study. If we think about what filters determine what types of bacteria are present in each community, think of the microclimate. So you can bet that the microclimate of a bird's mouth is going to be different than that of its colon, or at least I hope so, given the number of times they bite me. So that really does show in both of these graphs here. You have a little higher abundance of proteobacteria in the cloacal and fecal compared to the oral. There's a complete absence of Bacteroidota in the fecal. So there's a lot to learn here at some higher levels of taxonomy, looking at genuses and species as well. But it's a good overall description of the cardinal microbiome, at least in my population. Looking at differences among between the two sexes, males and females do significantly differ in their beta diversity. And if you look at the graph to the right, there's this column all the way on the right for U that denotes unknown sex. And it's not that I can't tell the difference between a male and the female. That's for when the birds are really young and they don't quite have that sexually specific plumage yet. And so it seems like there might be some differences between older birds and younger birds. And if you look at these graphs on this next slide, it turns out that relationship is trending towards significance. Once again, we have a relatively small sample size of youngins versus adults. So I'm hoping that my additional sampling from 2021 and whatever I catch this year will help bolster that sample size and clarify any differences that might be there. Because we don't really know how the microbiome develops across these animals' lifespan. And that can be really important depending on if the center gets in young birds versus old birds. If you think about it, most of the birds that they get in are during baby season, as they call it. So if there's a specific care that can be given to younger birds versus older birds, that could really make a difference. So there's a lot of cool relationships that we can see here. We found that birds with better body condition tended to have higher alpha diversity, more diverse microbiomes than those with poor condition. Same with the red beaks versus the orange beaks, and that males and females did have different microbiomes when it comes to their beta diversity. And then finally, there were the significant differences in both alpha and beta diversity among the 3 different microbiome types. So that really points out that you have to be looking at the right sort of system if you are going to be asking certain questions here. But most of the relationships that we saw were with the cloacal microbiome. Fecal microbiome didn't come out as significant that much because perhaps there was only 15 samples. And so the sample size just wasn't there. But cloacal and oral had the same sample size. So the fact that cloacal showed significant differences and oral did not, it really does lend some credence to all of this research interest in rodents and humans on the gut microbiome and less so on the oral microbiome. It seems like there is a tighter connection between the gut microbiome and the health of the organism. But there's some other things that didn't really come out in the stats. And for example, I sampled these birds throughout the year, and there was no seasonal trend in diversity. I looked at hematocrit, which is the packed cell volume, the number of red blood cells in each blood sample that I took. It's kind of an assessment of anemia and just general condition and health. That didn't show any relationship either. But that's kind of par for the course to have this big variation in things that do and don't come out in the stats, because we're still learning a lot. Like I said, this is the first Northern Cardinal microbiome study in existence, and it's one of only a handful in birds in general. So there's still a lot to learn and find out ways that the microbiome does actually link to the health of these hosts, because that's the actionable intelligence that we need, is to figure out how does it relate to the host. So clearly we have a lot to learn, but there are some encouraging kind of baseline relationships that we found here today. So that is about it that I've got. I honestly, I would not have gotten here alone, not even close. I'm not a microbiologist, I'm a field biologist. So without the help of people training me on those techniques, without all of the funding sources keeping me off the streets, I would not have gotten here today. So huge thank you to everyone that's been involved, to my co-authors and those that are keeping me going. With that, I will take any questions. For those that are following along on YouTube later on, feel free to shoot me an email if you have any burning questions. I'm happy to answer them.
Awesome. Thank you so much, Morgan. That was really good. So we can open the floor up for questions right now. I I did have a question, and I think you answered a lot of my questions already, but I was wondering if specifically alpha diversity was typically higher with the cloacal samples, and I found it pretty interesting that it wouldn't have been higher in the fecal samples, and I was wondering if you could touch on that.
Yeah, sure. So that's actually one of the biggest obstacles in microbiome research with birds is DNA yield. And that is how much DNA can you get out of a sample. So if you think about a fecal sample, which is a cohesive biomass turd versus a swab is going to most likely just be lower biomass than a fecal sample. And typically that will yield lower DNA. The fecal samples that I have, it was a small enough sample size that I'm not quite sure how representative that is of the entire population or species for that matter. But it did track that fecal sample had the higher DNA yield and diversity.
Awesome. Thank you so much. Yeah, so anybody else, if you want to unmute yourself, feel free and ask Morgan some questions and go ahead. Any questions are allowed here. You don't have to be, super scientific with those questions. If you just have a question about cardinals in general or birds, we are here.
I've got a question.
Sure.
I'm a veterinarian. I've been studying birds for the last 50 years. And the last 10 years, I've been studying the microbiome of birds through the use of the gram stain, and nothing as sophisticated as you. And then someone turned me on to a book by a lady called Stephanie Seneff, and I put it in the title of her book, Toxic Legacy, in the comments about the advertisement that you had for this. If the cardinals this is just my supposition, if the cardinals you're studying in that park are being influenced by spraying the grounds with glyphosate, that's totally changing their biome than, say, birds that are out in the middle of the Everglades. Have you heard anything about that?
I am not familiar with glyphosate. Can you give me a quick pitch on what that is?
Roundup.
Roundup? Oh, I'll have to talk to park management. I know that they spray for mosquitoes, but I believe that that's most of it. I will say that they don't spray in the areas that I'm working. It is a large open central grassland in the middle surrounded by woodlands. And the cardinals don't go to the big open parklands. They stick to the woods where it is completely unmanaged for any sort of invasive. That said, I know a thing or two about watershed, so it might be running off. I will definitely have to look into that. Thank you for pointing that out.
I think you'd find her book amazing, and I'd love to have a discussion with you and some other people on that in the future, because what they're feeding those birds when they're sick, if it has glyphosate in it, it's going to make their biome not respond the way it should. It's just an amazing subject. It's much deeper than I understand the biochemistry, but if you want to understand human, mammal, and avian physiology, read her book. It's just absolutely astounding.
Thanks for sending that. I'll definitely look into it, because the next biggest application to this, it's not a distinct passion of mine, but a lot of people that get into this avian microbiome field is looking at urbanization versus more of a ex-urban and rural area. So I'm sure that glyphosate is going to be a lot more of an issue in urban areas.
Well, the main thing that we've learned recently about it is the people who are running our agriculture system have discovered if you spray glyphosate on sugarcane, beets, potatoes, beans, you name it, the plant says, oh, I'm going to die. So it starts to put out more seed or more protein in the seed, or it makes the seed more nutritious. And so that makes the seed more valuable. So they now use it in sugarcane. It's just endless. And they call it a desiccant, but that's not what it is. It's not used to dry things out. It is used to kill the plant, but they're using it to enhance the value of the final product. And it's it's
definitely something to look into then. And I'm actually looking at my audience now, and I can see my buddy Akshay, who actually works with Roundup in the neuro department. So I'll definitely have to buy him a beer and see if he'll talk to me about some of the potential effects it could have. He looks at the effects on nervous system. So this is definitely really interesting.
What's his name?
Akshay Narain, if I'm butchering it properly.
Great. Excellent. Thank you.
Okay, thank you guys so much. Any other questions from our audience today? I have some more questions to keep it going, so if no one has any questions, let me know. Hi, Teresa.
Yes, my name is Teresa. I am from Green Bay, Wisconsin, and I rehabilitate cottontail rabbits to be released back into the wild. And I know we've had this discussion on birds. I'd like to know and ask you guys where I can find more information out about the microbiomes of rabbits, because that is the one thing that I have a very difficult time with is their gut health. Um, they really succumb very easily to having diarrhea and so forth. They have a very sensitive gut system, and I'd like to study more about how I can treat that those disorders in the rabbits, and maybe take care of that. Do you guys have any suggestions?
Being the academic that I am, I would turn to the research papers first in primary literature. So my approach would be to look on Google Scholar or any other PubMed search and see if there actually is research being done on this. And then you can either contact the authors of this. You might want to look in veterinary journals too, given that rabbits are commonly used as domesticated pets. And so there might be some lore on that. But the thing is, it doesn't have to be the microbiome specifically of that species. It could be rabbits in general, and that could be a nice stepping stone for you to learn more about that.
Okay, thank you so much for telling me.
Sure. I wish I could just answer the question, but yeah.
No, I definitely think that's a great start, especially Google Scholar. I use that all the time. It's a great tool where you can find tons of research papers on whatever specifically you might have an interest in. But of course, like Morgan was saying, there's a lot of research that still hasn't been done yet, more or less towards mammals, but not really in, I would say, the reptile and the avian field, as well as the fish field. There's just really not a ton of research in those areas. So I think that's kind of where the future is heading to, and hopefully we can get more people like Morgan out there that are interested in these things, so we can spread all of that knowledge to rehabbers like you, Teresa. And I know the wildlife center in your area, maybe their head veterinarian might have some references as well for some research that she might know about locally.
Right. Thank you so much. And I've enjoyed your presentation here today.
Thank you. I mean, the dream is to be able to answer that question and be able to say, yes, here, we have a pill for this. That might be 10 years off, but that is ultimately where this is going. Is we just got a red shoulder hawk. They are super stressed out classically. So let's give them the red shoulder hawk probiotic that will supplement their microbiome. There are actual technological applications to this research. We have to take our baby steps now, but cottontails could be on that list as well. Anything could be on that list.
And that's so amazing to think about, that you can just give a probiotic to an animal and it can affect their microbiome, which can affect their entire way on how they heal while they're at a rehab center. And then that's also similar and simulated in human science as well. So it's really on the edge of research currently.
Right. I also don't want to be, like peddling magic here though, because I doubt it will ever, unfortunately, be that simple. But that actually reminds me, Teresa, there is a lot of research on, if you just simply provide soil and environments that is from the native habitat of whatever the patient is, that can actually normalize a lot of the issues in the microbiome. They've actually done research studies on that in a lot of salamanders. They'll give them soil from their home turf, and that will pretty much normalize their microbiome when they're in human care. So there could be something for that.
Right, exactly. And it's unfortunate that when we do get these rabbits that are brought in, sometimes the humans have tried to feed them, and that's why it's important that we ask them if the rescuer has fed anything to the animal. It's
Right.
very important. I did get a batch one time that they were feeding the rabbits, and these are all rabbits that are um, under a week old when I get them, and they were feeding them cow's milk, and I mean, it's
House.
amazing, but of course the general public is not um, educated with this. That's why we prefer that they just kind of leave the nests alone. A
Mhm.
lot of times they bring them in because they think that the mother has deserted them and so forth. And yeah, it's an education process to the general public for sure. We try to keep them as much educated about it as we can, because we do want all the wildlife to remain wild. Right?
Agreed.
Yes,
Agreed. Yes.
definitely, Teresa.
Thank you so much for your interest.
Yes.
So
Thank
we
you.
got two more questions so far. So I have Hans first, and then Joseph will come to you in a second. So Hans asks, do you know if the singing behavior of these birds are affected by their microbiome?
Ask me again in May. That is actually the focus of my current research that I'll be leading a team of undergrads working on this spring. So while I'm out there catching the birds and taking my samples, they're going to be out there recording the same birds and seeing not only are there broad correlations with the way that they sing in the microbiome, but also does that change with me capturing them and releasing them. But there is one of the metrics that we typically look at in our bioacoustics lab, and that's called vocal performance. It's essentially how good of a singer is that bird. And this can vary wildly from one individual to the next. And so that vocal performance is thought to be the thing that females will key in on when they're listening to the male sing his beautiful song. And it might also be something that other males listen to when they're thinking about picking a fight. So this could actually have huge consequences for fitness, and therefore the microbiome might play a role, especially because of that gut-brain axis between the microbiome and the brain. And song is a learned behavior. So if the bird doesn't have a strong brain, if it doesn't grow one when it's a kid, then it might not be able to sing all that well when it's actually, research does show it will not be able to sing well when it's older. So if the microbiome plays any role in that, it'd be really interesting to know.
Thank you, Morgan. Okay, we'll go on to Joseph if you want to.
Hey, Joe.
Hey, Morgan. How's it going?
Good to see you, buddy.
My question has to do with your relationships that you saw with the beak color and the feather color and relating to the diversity. So was that with just males, or was that with males and females together, or did you see any relations to see if that it was specifically found in females different than males?
Right. So the beak coloration, that was for males and females.
Okay.
And surprisingly, beak color didn't differ all that much between males and females, at least in the samples that I took. So I did not look at them separately when doing that analysis, because I know that's what you're driving at. But they're so for beak coloration, yeah, it was males and females that that show that relationship. And the interesting thing about the past research with beak coloration cardinals is it's actually the beak coloration of both of the birds that are involved in that assortative mating. It's not just the female checking out the male, but also the male checking out the female. So I did, however, only take the back feathers from the male. They're the only ones that have that bright red back. I didn't take any feather samples from females. So that is only in males.
Okay, cool.
Thank you.
Well, thanks. Good talk.
Appreciate it.
Okay, I had another question for you, Morgan. So
Yeah.
I know you mentioned briefly about how like seasonal changes that didn't show to be very significant in your data. However, migratory changes, did you notice anything different with cardinals like migrating? I know our population in Florida is a little bit different, but just was wondering if that's something that you saw.
Yeah, so our population doesn't migrate, so that wouldn't be something that I could look at in ours. But I will say that for any of those in the audience that are interested in migration in the microbiome, it's actually a really hot topic, because if you think about it, the microbiome is going to be susceptible to any current biochemistry and physiological changes that the host is undergoing. And for those that know a thing or two about migration, the body changes dramatically throughout the different stages of migration as they're putting on, huge fat reserves. They're if they're on the come down after the breeding season, their gonads and sexual organs will be smaller and changing the biochemistry and hormone levels in the body. So all of these things do fluctuate with stopovers and wintering. I don't know anyone working on that. And cardinals wouldn't be the birds to look at. But yeah, I was actually surprised that there wasn't any change in it throughout the season, though, because hormone levels do fluctuate even though they don't migrate.
Mm-hmm. Interesting. Well, thank you. Thank you. So anybody else have any questions so far, or think of anything while we were talking about other questions? You can either put it in the chat, raise your hand, unmute yourself, either or anything is welcome. Okay, I think we're all good then. Well, Morgan, did you maybe want to touch on anything else before closing up?
Sure. I'll fly through the last couple slides that I had on as backup just while everyone's here.
We had a lot of thank yous in the comments. So everyone said your presentation went awesome, and they said thank you for sharing.
Thank you. No, it's my pleasure. So yeah, there's some other things I am involved in to answer Hans's question. We do a lot of bioacoustics research in my lab, and one of the things that we're trying to figure out is how good of a singer is each bird. And we can do this by recording the birds. We use a little parabolic dish, and we find a male that's singing. Females will sing as well. So we will find a male or female that's singing, and we will record that song with a microphone that records it onto basically a big Walkman. It's a lot more advanced than that, but that's basically what it is. And that can then be fed into software called Raven Pro, and that allows us to see what the song sounds like. Say that again, just to wrap your head around it. We can see what the song sounds like. So we can graph it out. We have frequency or pitch on the y-axis over time on the x-axis. And if I'll crank my volume, you can hear what this song sounds like. Is that coming through in the speakers?
Yeah, it came through on my end.
Okay, cool. And so cardinals are one of those species of songbird that sing multiple types of song. When they're young, they learn not just the one song type, but they learn a bunch of different song types. So there's a lot to be learned about, why might they learn multiple different song types? So this is actually a separate song type sung by the same individual that sung this one. So I'll play this one once more for your memory. And now this one. So it is, that was my only way to cut the sound off. So it is wildly different between these two different song types. And there's a lot of metrics that we can take to allude to Hans's question of, does it correlate with the microbiome? We can look at vocal performance. How many individual trills are they forcing into each little increment of time? There's this thing called frequency modulation. How much is the frequency changing per second? How many notes per song? How many different song types do they know? There's a lot to learn here, and I don't know of anyone that's actually testing it relative to microbiome samples. So I'm hoping to have the answers to that after the spring. And I just wanted to accentuate the knowledge gap that exists. This is a slide I usually like to show, but I didn't quite have time for it. This is the relative research interest of avian microbiomes compared to that of other animals. So mammals is, this is number of papers since 1980. So mammals, mostly lab rats, is at the top, followed pretty distantly by humans. And then at the very bottom, we have domestic birds, which is mostly chickens and turkey, because humans care about that economic interest of poultry. And painting the x-axis is a dotted red line you can't see, and that's wild birds. It's almost nothing going on. And this is, as of 2016. That has changed actually pretty significantly in the last 6 years or so. But there's still a lot more that we need to do to catch up with what's going on up here. And then for anyone in the audience that was interested in a bunch of different ways to identify Northern Cardinal, I work with males. I have the honor of working with the angry females as well, and the teenagers. They're trying to get their pretty red feathers as well. And then occasionally I'm able to catch nestlings if I find a nest, which basically don't look like cardinals or birds for that matter. So there's a lot of interesting work to be done looking at how the microbiome develops over time. And that, I believe, is everything I have.
Awesome. Thank you for sharing that additional information. I think it definitely, helps a lot of people. They've come from a bunch of different backgrounds, so you really never know if you're getting someone that this is their first introduction into birds in general or cardinals. Some people might have never even seen a cardinal before. So it's definitely interesting to share this research with so many different people. But then also on the other side, we have a bunch of other professionals in the field, as I've noticed, that have been kind of tuning into some of these more advanced questions. So thank you so much again for sharing your presentation. We got some thank yous in the chats as well. And yeah, I look forward to definitely, hooking you up with anybody who might have any further questions later on, so that they can have access to your email if they might think of anything. I know I always think of something when the presentation ends and I'm like, oh, I definitely should have asked that person. But yeah, so we will definitely share that information later on. But thank you so much, Morgan. And that concludes everything.
Oh, my pleasure. Thanks so much for having me. Keep up the good work at the center.
You too. We need more bird researchers out there.
Absolutely.
Awesome. Thank you, everyone.